Deanna Fournier here, Executive Director of the Histiocytosis Association, and I am very excited to be joined by special guests today to discuss a new collaboration between the Innovative Genomics Institute, or we might refer to it throughout our conversation as IGI, and the Danaher Corporation, a global life sciences and diagnostics innovator. So the Danaher-IGI Beacon for CRISPR Cures Center is what we'll be talking about today, which uses genome editing to address hundreds of diseases, including rare genetic disorders that have no cure. And experts from UCSF, UCLA, and UC Berkeley are some of the institutions that make up the IGI team. And so we have three very special guests with us today, and I'd love for each of you to introduce yourselves to our audience, and then we're going to learn a little bit more about the CRISPR platform and IGI and how this applies to histiocytosis. So Dr. Hermiston, we'll start with you.
Thank you, Deanna, for the opportunity to speak with our colleagues, patients, and families today about this very exciting collaboration. My name is Dr. Michelle Hermiston. I'm a pediatric oncologist and transplant physician at the University of California in San Francisco, where I've had a very long-standing passion to cure patients with hemophagocytic lymphohistiocytosis, or HLH, and also direct our pediatric cell therapy program. And so I'm very excited when there are opportunities to translate novel therapies into care for our patients.
Hi, everybody. I'm David Nguyen. I'm also a physician at UCSF in infectious diseases, adult infectious diseases, and a physician scientist and engineer by training. I'm jointly appointed at Berkeley in immunology, and this is where my research lab is. And I do benchtop work, try to translate our technologies into cures for patients with rare immunologic diseases, which is my passion. I see patients with immunodeficiencies at UCSF, and then I work on trying to understand these mutations that cause disease and cure them over at Berkeley in the Innovative Genomics Institute.
And my name is very fortunate in the sense that I get to work with amazing physicians like Dr. Hermiston and Dr. Nguyen, and also be a scientist at the Innovative Genomics Institute that was founded by Jennifer Doudna with a vision to make CRISPR gene editing a standard of clinical care in certain diseases. My actual name is Fyodor Urnov. I'm a professor here at Cal, go Bears. But really our partnership with UCSF is essential for what we're trying to do, because we're a bunch of researchers, and the patients and the unmet need is, of course, right across the bay. I'm delighted to be given the responsibility to be the overall director of our remarkable new partnership with Danaher, but I really want to emphasize that the beating heart of the project are the patients in Dr. Hermiston's and Dr. Nguyen's practice, and the remarkable science that we're going to have to put together in order to deliver a CRISPR treatment for this devastating disease. Very happy to be here.
Thank you, and we're just so grateful for the three of you taking the time out of your, your busy schedules to sit down and have this conversation. Because my understanding is it's a very exciting opportunity, and specifically starting with the HLH, disease group and for the HLH community, but it may also have implications down the line for other histiocytic disorders. So can you tell us a little bit more about the collaboration that exists and what the CRISPR platform will offer to rare disease?
2023 was a special year for everyone working in this space, because the world gained its first ever gene editing medicine, in fact, two, for prevalent diseases. And the HLH community doubtless knows that these medicines were for sickle cell disease and beta thalassemia, so disorders of the blood. What does that mean in English? It means that you can gene edit a human being's hematopoiesis to a curative outcome. So all of us, physicians who treat patients, scientists who build CRISPR, have one question: what will be the next 10 diseases cured? And I don't want to be Debbie Downer, but I want to be Debbie Realist. The for-profit sector is sputtering a little bit right now as it's trying to figure out its business model of how it can build a medicine, not just for sickle cell disease, which is amazing, of course, but it has 20,000 patients with severe disease in the United States. That's a formidable patient population to commercialize a medicine for. What about a rare disorder of the immune system? Again, something Dr. Nguyen works on, which 20, 30 patients in the US. How do you make that into a medicine? And that's really the centerpiece of what we are trying to do in partnership with Danaher. How to build a fundamentally new model of diagnosing a patient with a severe disease, let's say a child who arrives in Dr. Hermiston's practice, and then take that diagnosis and engineer a CRISPR medicine. And this is something Dr. Nguyen runs a lab on, he can speak to, and then figure out a way to manufacture a cell therapy product for that child before that child, frankly, succumbs to their condition. And do so in an interaction with the regulators where, the FDA will understand that we have to move quickly. We don't really have time to spare. And ultimately administer this therapy to the child in a way that's cost affordable, because right now these medicines are $2 to $3 million. So we are very grateful to our partners at Danaher for sharing the same sincerely heartfelt mission of doing the right thing. Danaher is a corporate giant, frankly. They provide tools and solutions to the life sciences and biotechnology industry across pretty much everything. They're a little bit of a one-stop shop for so many of the bits and pieces we need. And the fact that Danaher so graciously partnered with us and supported our teams to take gene editing for, a very important genetic condition. But frankly, let's be clear, this is not like major cancer or heart disease. And yet here we are, a major research nonprofit institution on the UC Berkeley campus, partnered with elite physicians at UCSF and UCLA in partnership with a major corporation, Danaher, working towards precisely that, a CRISPR treatment for HLH. A pretty remarkable moment, and one we're delighted to be at the start of.
It excites me to hear you say this. I think one of the challenges is that HLH can be caused by mutations in a wide number of genes, which makes coming up with a cure challenging. The standard of care currently is to try to calm the inflammation, and then for children and adults who have a mutation, we replace their entire hematopoietic stem cell system with a bone marrow transplant, which comes with high level of toxicity, exposure to a lot of drugs, and the potential for a lot of complications. The beauty of CRISPR-Cas gene editing is that there's a hope that we'll be able to do this in a way that is much less toxic to the patient. And the hope is to be able to develop a platform so that we can use the same method, but target different genes by developing different guides, so that this then becomes accessible to the, N of 1 or N of 2 patients who have this rare disease, but we can use a common approach to provide cures that are hopefully much less toxic and safe for patients.
Yeah, from the engineering translational perspective, and the goal is, we see these patients in the clinic and we're like, in theory, this shouldn't be that difficult. Like, we can actually go back to the lab and show that we can do this in bits and pieces in tiny test tubes. That's very different from doing it in, like a human being in a clinical trial and trying to cure a patient. So how do we get there? And I think Michelle hit on one of the aspects of that, and that's this concept of platform. And the amazing thing about CRISPR is that it's so versatile. It's a tool. It's like but it's not an Allen wrench. It's not a screwdriver. It's more like that multi-purpose tool where you can change the head on your screwdriver to D Phillips or to flathead or to Allen wrench or to all those weird ones that come on, all the other computers and things like that. And the idea is that you can repurpose that tool very easily. And so when you look at a disease like familial HLH, where there's many different genes and each patient doesn't have the same mutation, is it N of a couple of hundred, or is it N of 1? Because one patient might have a mutation that's never shown up before. So one of the patients that got me first interested in this is a patient who had 2 different mutations in one gene that had only one of which had ever been seen before in any other patient. So how do we even know if those mutations are actually causing their disease of HLH? And so the first thing that we do in our lab is use CRISPR gene editing to say, well, is it broken? Is that how it's broken? And that's important if you're trying to fix it. If you don't know it's broken, you can't actually fix it and correct it. And so that's a new concept for the rare diseases and mutations, that the first thing you need to do is validate that that's exactly the mutation that's causing this patient's disease, particularly when there could be a mutation anywhere in the gene. And the second step is, how do you actually fix it? And that's the part where the platform is really awesome, in the sense that if we figure out how to do it for 1 or 2 or maybe 4 different mutations and say, look, in these 4 different mutations, this works, all applicable in the same gene or in a couple of different genes for HLH. The idea is then all we have to tweak a little bit is the bases and the code and the sequence and the guide RNA, what's called the guide RNA, that tells the CRISPR enzyme where to target in the genome. And once we retarget it, it should have the exact same effect or very similar effect, which is to say a therapeutic outcome. And so that's what we mean by we say platform. The idea is that we'll be able to say, anybody who has this disease state in any number of genes that might have caused that disease state and any different mutation, we already know that we can retarget pretty quickly. We can just change the screwdriver bit just a little bit so that we get to the right spot, the right target, and correct the mutation in that patient's own cells, which is very different from giving them the same small molecule drug that everybody else is getting. And so that's the goal, that's the promise, that's the hope of the platformizable aspect of CRISPR gene editing.
Thank you. It's very exciting. I mean, it touches on a couple things for me. It's a bit of that precision and personalized medicine that we're hearing so much about now. But also, you know, I think about, you mentioned this N of 1, N of 2, these small patient populations, which often create this prohibitive situation for rare disease drug development, where, you know, we see it could take 10 or more years to have a therapy make it through to clinical trial to be able to be used by patients. And even the clinical trials often sit for a long time trying to recruit and enroll. So this is really exciting. It sounds like it has a lot of potential impact for HLH. And maybe you could talk a little bit more about you know, the treatment and research potential here. I know you touched on it a bit, but specifically for HLH in the field of histiocytosis, what this can do.
My hope is that this is really the first step in, you know, a number of different therapies that will improve outcomes and treatment for our patients. One of the things that we have been discussing is to be able to edit the T cells as a first step to tide a patient over, and then to be able to use that same approach, but edit and essentially fix the specific mutation that's in their hematopoietic stem cells. So really a two-step process where you could improve things. There was a very interesting paper that was just published this past week that shows that editing the T cells can actually work in a mouse model of HLH, where the mice have engineered a mutation in perforin, one of the most common mutations that we see in patients with HLH. And they gave the mice HLH by infecting them with the virus, and then gave T cells and could actually cure the mice and prolong their survival. And so that's an excellent mouse model of what we hope to actually be able, through this amazing collaboration of multiple stakeholders, and to be able to really take this to patients in a way that will be adaptable to whatever mutation the patient, or two mutations, as David mentioned, might have. And maybe, David, do you want to expound on how you think about this from the science side?
Yeah. So, and I think this is like, we have this opportunity to do something that hasn't been done before. And there's a couple of things that we're going to try to do that haven't been done before. And I think the first is to treat patients with their own T cells, right? So in HLH, that really hasn't been done before. And it's a critical part of the problem specifically in the HLH disease. The T cells and the NK cells are dysfunctional, and that's a root of the problem. And so there seems to be pretty good evidence that fixing at least the T cells can actually ameliorate the disease, if not provide some medium to long-term benefits. We're very hopeful that that's what's going to happen, and so that's novel. Nobody's done that before, and so that's one of the innovations that we're looking at here. And it's a great approach because CRISPR engineering in T cells is probably the most advanced engineering, even though the first approval is in hematopoietic stem cells. The efficiency is actually higher in T cells. So we have I think we have a great shot at this, and I think that that's going to benefit patients. Second, we are taking, as I mentioned before, this platform approach, and that actually has never been cleared by the FDA, a platform approach to editing where we won't know ahead of time which patient mutations are going to show up in the clinic. And so we'll have ready all of the medical capabilities and manufacturing capabilities to create a individualized gene edit medicine for that patient. And this is going from even precision medicine to exact medicine. That's how I like to think about it. So this is an exact medicine approach. And the goal and the innovation here is to try and prove that we can do this. And we think we have all the tools to do it. And B, to prove to not just the FDA, but to patients who ultimately decide what medicine they're going to take, and to the clinicians, that are going to help administer those medicines, that this is not only a safe approach, but a robust approach. And then we can get essentially the green light, hopefully through a clinical trial and through the FDA, to say, look, this has never been done before. You have this tool set, but nobody's ever deployed a tool set as a medicine. And so we're going to try and deploy a tool set that can be individualized to each patient. And I think ultimately what that does not only benefits in HLH, but once we've been able to show that we can do this, this benefits all rare diseases where the mutation is never going to be exactly the same, but the approach could be very similar. And so I think that that's an additional layer of innovation that is, exciting and daunting, but we feel, the group that we've, assembled can do all of this. And so I think, yes, we're approaching HLH, but could it help with other histiocyte disorders as we learn more about the genetic underpinnings of those? Absolutely. Even proving that some of these histiocyte disorders can be treated with T-cell alone approaches potentially. So I think there's a lot of potential benefits that go beyond the collection of syndromes of familial HLH.
This is a good opportunity for me to simply say, we as developers of the tools are just in awe of the patients who agree to be subjects on clinical trials, right? Like ultimately these human beings agree to be part of an experiment. It's clinical research. And I just want to say, as somebody whose job is much simpler than Dr. Hermiston's or Dr. Nguyen's, which is I just got to deal with little test tubes. Nobody knows if this is going to be safe and effective. The only way to find out is do a clinical trial. But based on everything we've learned in the past, basically 20 years of genetically engineering human cells using gene therapy, using engineering T cells for cancer and other treatments, and then, of course, the more recent CRISPR success with sickle cell diseases, now is the right time in the lifespan of this technology to be swinging at this. This is not, in other words, this is well thought out. I want to really credit Dr. Nguyen and Dr. Hermiston for their leadership in showcasing HLH as the disease to go after. We had this meeting here in this building saying, well, if we partner with Danaher, what's a disease that's A, severe, B, nobody is working on genetic therapies for it, even though they should, and C, where we stand a meaningful chance. And then the physicians said, have you heard of HLH? And I will admit, I was one of the researchers in the room who said, I have not. I was blushing, and here we are.
And I'll say that it's not just the two or three of us that are here on this call. There's a lot of other people who've been involved in thinking this through. So.
No, this is definitely going to take a team. I think we're, and a partnership with a corporation like Danaher that has the resources and also the passion and the commitment to equity. These are diseases that, you're unlucky if you happen to be afflicted with HLH. The science is there in the lab, and this is, I think, just a really unique and exciting way to merge all of this fantastic research, the engineering in the lab, and the technology that we need to actually be able to bring these things to patients. And so it will be stay tuned. We have a very aggressive timeline to try to get this to through development to the FDA and ultimately to patients. But it is a privilege and extremely exciting to be able to try to do this for our patients.
That the approach we're taking, CRISPR gene editing, the enzymes don't care what you look like. You look all the same at the genetic level: A's, C's, G's, and T's. One of the patients that I got inspired by was a patient who, because of their ethnicity, was not able to find a bone marrow transplant, or at least a low risk bone marrow transplant, because they had low representation in the publicly available bone marrow databases. This is an example where gene editing can really bring health equity and really bring access to patients that never had it before to the other, methodologies of treatment. Of course, these things aren't cheap, and that's why companies are involved, right? And that's why we're really glad to have a place, an institution, I would even call it, like Danaher, to help make this happen. But an overall goal of the Innovative Genomics Institute, of Berkeley, of UCSF, and of all of us is to make this as accessible as possible. So as we engineer these things, as we design these things, as we think about these trials, the goal is to make this, as affordable as possible, as accessible as possible. And ultimately, this is not the first pass at this. We have so many other, things that we're working on that will go beyond this initial Danaher Beacon towards novel technologies that will allow us to do that in such a way that it's every year getting cheaper, every year getting more accessible. And I think ultimately that is one of the ultimate benefits of using a tool, that you can figure out better ways to deploy it.
I have chills because I'm hearing things that are so exciting and create a lot of hope for the histiocytosis and rare disease community, putting the power, empowering the patient family community to make a decision that feels good. Affordability, thinking about health equity and reaching communities that sometimes don't have access to treatment, even just across the map. I'm even thinking about people that we connect with who have, for example, like the XIAP2 mutation. There's very few of them, and finding an appropriate therapy that's going to help them, that's not bone marrow transplant. Some patients aren't eligible for that. This is just so exciting and creates so much hope. And the fact that you mentioned there's an aggressive timeline, I know that there's a lot of other work that you're doing, and so the focus and dedication to getting this sort of sounds a bit expedited to the FDA because you believe in it so strongly is really exciting. I can't wait to learn more as you go. I mean, I'm thrilled. And I think that this is really the future of medicine, and especially for conditions like HLH, to hear that there could be potential for it to cure and it be a safe and effective way to do that is minimally invasive it's incredible. Is there anything else that you'd like to share about the project, or just any other messages that you wanted to share with the Histio community?
Well, I think if I may ask my clinicians on the call, I just got an email from a patient, and they said, for a different disease. And they said, look, my child has this genetic condition. I saw your interview on NPR. What are my options here? So can I ask the clinicians on the call, like, how does a patient whose life or whose family is affected by HLH think about the possibility of becoming part of this effort?
Oh, great question. I think research is what drives cures, and so I think at this point there are opportunities for patients. The FDA has a lot of requirements. We need to develop this, and we need to do it in a way that is safe. And so I think we're still a bit off from being able to take this, overnight, and the patients with this disease need it now. And I think that's what drives and motivates us, but it's going to be a bit, though I think in the meantime, something that can be extremely helpful is for us to develop a broad bank of biosamples from these patients. So that patients are able to biobank their specimens when they're getting a blood. We don't procedures just to get blood samples or bone marrow samples, but if they're having a test done, they can choose to, under informed consent, donate a tube of blood or a sample of bone marrow to the NACHO Consortium, which has a biobanking freezing protocol. We also have a second protocol through UCSF if families want to, and through the IGI if families want to donate tissue. But that's incredibly helpful for helping us get to that goal of engineering solutions on a per patient basis.
As a clinician who sees patients with rare diseases on a regular basis, we get this question a lot. And I have patients with other diseases right now who are sitting there thinking, hey, can I wait for a gene editing cure, or do I need to do something else more standard of care at the current? And I think that's really a conversation with the expert physicians who are taking care of you. And that's for HLH in particular, there, as we progress in this, there will be more options, but that option's not there yet, right? It takes time. And we are at the very beginning of this and the start of this. And so I think that it's we have the goal of getting there as quickly as possible. And I think that the idea of this taking 10 years is probably not where we want to be. We want to be at the door much quicker than that. And ultimately, this is something that is conversation with the community, with the FDA, with the other agencies that are involved here in figuring out how we're going to do this safely and effectively, right? to get it as quickly as possible to our patients. Participating in other clinical trials is always a great thing to do if you can and if you feel comfortable and if you understand what you're doing. And giving access to tissues through biobanks is always helpful because you never know when that's going to get used by somebody else who can help even more. And I think that ultimately one aim we can have is to offer some hope for the future. Even if we don't have it like in the immediate right now.
Well, this is fantastic. And I hope that we can reconnect in a year's time or maybe even less as you have milestones. I'd love to share those milestones with the community so that they can see really what you're learning, because I think there's so much that will happen in a short amount of time that will just build on itself. And it's just really very exciting to know that we're at this point, and so grateful that HLH was brought forward for this opportunity. And I know that everything you described is going to bring a lot of hope to the community, and just see what happens in the next couple of years and what we are able to learn.
The training wheels are off. This is not, this is not a dress rehearsal. We are in it. This is the real thing. So I think that this note of, legitimate optimism that David just provided, I think we would not sign up for this if we were not prepared to approach this with a note of healthy optimism. So please watch this space, and thank you for the opportunity to speak with you.